The purpose of this video is to give you a background in hematopoiesis and its malignancies that include both lymphomas and leukemias. Basically, this is what you need to know to understand this rather complex topic. I am going to start out with some simple definitions. Hematopoiesis means the production and development of the formed elements of the blood, and hematopoietic malignancies represents the unregulated clonal proliferation of the hematopoietic stem cells. This includes a number of different cell lineages, since there are a number of different formed elements in the blood. This slide shows the formed elements of the blood. Now I use the term formed elements here instead of cells, because technically platelets are not cells, but rather fragments of cells. They are derived from megakaryocytes in the bone marrow. Also, there is some question as to whether erythrocytes should be considered to be cells, since they lack nuclei. Be that as it may, in addition to platelets and erythrocytes, the blood also contains leukocytes, or white blood cells, of which there are 5 types: monocytes, lymphocytes, neutrophils, eosinophils, and basophils. Now, all of the leukocytes are involved in one way or another in immune surveillance of the body. That is, they help protect the body against infection. Now, there are a couple of features I would like to emphasize. First, the 3 leukocytes shown on the bottom here, that is the neutrophil, the eosinophil, and the basophil, all have small granules in their cytoplasm, and for this reason they are also referred to as granulocytes. You should also note that their nuclei are highly lobulated, and for this reason they are also known as polymorphonuclear leukocytes, or polys for short. This term particularly applies to the neutrophil. And secondly, I would like to emphasize the importance of the lymphocytes. These are the real stars of the immune system. These cells have a slightly different developmental history than the other formed elements, as I will describe later. Now, the leukocytes represent only a small fraction of the volume of the blood, far less than that of the complement of the erythrocytes. In contrast to the platelets and erythrocytes, the leukocytes spend only a relatively brief period of time circulating in the blood, on the order of hours or perhaps several days. They then enter the connective tissue where they take up residence. Now, all of these formed elements of the blood are constantly being turned over and must be replenished. This is the process of hematopoiesis. This slide shows the lineages of the different formed elements of the blood. On the far right, under the heading of mature cells, you can see the different cells are easily distinguished from one another. For example, the lymphocytes and the erythrocytes. Now, the lymphopoietic lineage give rise to the lymphocytes, and the erythropoietic lineage gives rise to the erythrocytes, and the granulocytic lineage gives rise to the granulocytes. I think you get the idea. However, as you trace the different lineages back, that is, you go to the left in this diagram, following the headings precursor cell to progenitor cell, and finally the stem cell, you will note that the cells lose their distinguishing features and begin to resemble each other. Furthermore, as you go back, the different lineages converge with each other. And if you go far enough back, then all the different lineages come together at the pluripotential stem cell, as indicated here by the red box. Now, the pluripotential stem cell has the capacity to give rise to all of the formed elements of the blood. And like other stem cells, when it divides, one daughter cell replenishes itself, while the other starts to undergo terminal differentiation. These pluripotential stem cells reside predominantly in the bone marrow. Very early on in hematopoiesis, the pluripotential stem cell gives rise to 2 major divisions: the lymphoid multipotential stem cell and the myeloid multipotential stem cell. Here's where it gets a little bit confusing. Some, but not all, of the lymphoid multipotential stem cells migrate out of the bone marrow and enter the lymphoid organs. This figure is somewhat misleading because it shows all of the lymphoid multipotential stem cells migrating to the lymphoid organs. In fact, only those that give rise to the T lymphocytes migrate out to the thymus, while those that give rise to the B lymphocytes remain in the bone marrow. This diagram illustrates the hematopoietic organs. This includes the bone marrow as well as the lymphoid organs of the thymus, the lymph nodes, and the spleen. As shown in the bottom center here, the lineage that gives rise to the T lymphocytes migrate to the thymus, while those of the B lymphocytes remain in the bone marrow. After final maturation of these lymphocytes in these two regions, they can then migrate to other lymphoid organs, such as the lymph nodes and the spleen. The pluripotential stem cell also gives rise to other lineages that remain in the bone marrow, and these are collectively referred to as the myeloid multipotential stem cells. Now, the term myeloid simply means bone marrow. So this includes the lineages that give rise to the erythrocytes, megakaryocytes, and the leukocytes, with the exception of the T lymphocytes. Now, for historical reasons, hematopoietic malignancies have been classified into two major groups, lymphomas and leukemias. Lymphomas appear in lymphoid tissue, that is, the lymph nodes, the thymus, or the spleen, and they arise from neoplastic lymphocytes. They can also involve the blood and the bone marrow, and in some cases resemble leukemia. On the other hand, leukemias appear in the bone marrow. They can arise from either lymphoid or myeloid stem cells and can replace the normal hematopoietic tissues in the bone marrow. Often, leukemia patients present with anemia or bleeding disorders because they are not producing sufficient amounts of erythrocytes or platelets. This slide just illustrates this point. For lymphomas, think lymphoid organs, and for leukemias, think bone marrow. Now, there is a problem with this old approach for classifying hematopoietic tumors. It is based more on the location than it is on the lineage. In addition, there is considerable overlap between lymphomas and leukemias. The lymphoid cells can move around and involve both the bone marrow and the lymphoid organs. A superior method for classifying these malignancies would be to identify the particular lineage and the stage that it is at in the developmental sequence. However, this is hard to do just using regular staining, since the cells look very much alike. This is where the CD system for identifying cells has come into play. CD stands for Cluster of Differentiation or Cluster Designation, and it was established by an international workshop in 1982. Basically, what it does is identify different families of proteins on the surface of leukocytes and other cells. The proteins are identified through monoclonal antibodies which recognize specific structures within the proteins. For each particular CD, there are two different monoclonal antibodies that can be used to recognize its structure. Generally, these are mouse monoclonal antibodies that specifically bind to proteins on the surface of human leukocytes. Now, at present, there are over 350 different CDs that have been identified. And by examining the combination of CD markers, it is possible to identify the lineage of the cell and the stage of its development. Now, this slide shows the immunotyping of the lymphocytic lineage. However, the same principle applies to other lineages, such as those that form the erythrocytes and the granulocytes. On the far left, you can see the lymphoid stem cells express the marker CD34. This then separates into the B cell lineage on the top that remains in the bone marrow, and the T cell lineage on the bottom that migrates to the thymus. As the cells mature into functional B and T lymphocytes, the expression pattern of the CD markers changes. If you look at the thymocytes on the bottom, you can see that the CD34 marker is eventually lost and others appear, For example, the common thymocyte expresses CD1, 2, 4, 5, 7, and 8. Thus, by analyzing the tumor cells for these CD markers, it is possible to determine what lineage the cell belongs to, and where in this developmental progression the tumor cells have become arrested. Now, the final topic I would like to address is the structure of the bone marrow and the lymph nodes, since you will be examining both of these in the laboratory. This slide shows a view of the bone marrow. Now, the best way to understand its structure is to follow the blood supply. You can see that the nutrient artery enters the bone midshaft. It splits and then runs towards either end. As it does so, it gives off collaterals that carry the blood towards the inner edge of the bone. The blood then reverses direction and then migrates through the sinusoids of the bone marrow towards the central marrow vein and finally out of the bone. Now, as the blood flows through the sinusoidal capillaries in the marrow, the newly matured formed elements of the blood enter the circulation. This is a thin section of the bone marrow. The large spaces are where the adipose cells had resided. However, the fat that they once contained has been extracted during the processing for histology. The rest of the specimen consists of the hematopoietic component. The most obvious structures are the megakaryocytes that give rise to the platelets. These cells are very large and multinucleated. Also observe nests of erythropoiesis, where the red blood cells are being made, here labeled as erythroid islands. These are characterized by the presence of small, round, condensed nuclei. Immediately after this stage, the nuclei are ejected from the cells, and the erythrocytes undergo the final stages of maturation and enter the blood. And finally, there are regions of leukocyte maturation, here labeled granulocytic precursors. You can tell that these cells are leukocytes because the nuclei are highly lobulated. Now, there is one important detail that I should mention. As a person ages, the hematopoietic component of the bone marrow is replaced by increasing proportion of adipose tissue. This is the red versus the yellow bone marrow. By the time a person has reached adulthood, the hematopoietic component is restricted to just the flat bones. Consequently, in an adult, a bone marrow biopsy is most often taken from the iliac crest of the hip, which is flat and still contains hematopoietic tissue. Now, the other structure I want to describe is the lymph node. The function of the lymph node is to filter the lymph. The lymph is the fluid that collects in the tissues and is channeled into lymph vessels throughout the body. Significantly, it may contain antigens, such as bacteria or whatever. All of the lymph passes through at least one lymph node prior to being returned to the blood. Perhaps the best way to describe the structure of the node is to follow the pathway of the lymph. The lymph enters the node through the afferent, that's afferent with an A, lymphatics. It flows through the subcapsular sinus and down the trabeculae and finally exits the lymph node through the efferent, that's with an E, the efferent lymphatics. As the lymph percolates through the node, any foreign substance that it contains, such as bacteria, are picked up by the macrophages and other cells, which are collectively known as antigen-presenting cells. These cells ingest the foreign material, break it down into small pieces, and place the pieces of foreign material on their surfaces. This is then presented to the surrounding lymphocytes. If the lymphocytes recognize the antigen, then they are activated. They migrate to one of the germinal centers, undergo expansion, and begin to divide. In conclusion, I hope that this brief introduction allows you to better understand the rather complex nature of hematopoietic malignancies.